My name is Amanda Therrien. I am an institute scientist at the Moss Rehabilitation Research Institute, where I direct the Sensorimotor Learning Laboratory. My research is interested in mechanisms through which your nervous system integrates sensory information like vision, what you see, and proprioception, what you feel, with motor commands to control your movement and learn to make new movements. One line of research in the lab is trying to understand whether we can improve rehabilitation interventions for a movement disorder called ataxia. Ataxia is the result of damage to a brain structure called the cerebellum and presents as impaired movement coordination. The cerebellum is crucial for a process called sensory prediction. And we need sensory prediction because the processing of sensory information from the body is actually quite slow. So for example, everything you're seeing right now actually happened in the past. Depending on your nervous system, it happened about 120 milliseconds ago. While this may not seem like a very long time, it's actually problematic for movement control. In the case of ataxia, where sensory prediction is impaired, their motor system has to rely on this time-delayed sensory feedback. This forces movement control to have to operate through a kind of negative feedback loop, which produces oscillating behavior. And this is what we see with ataxic tremor. The goal of this line of research is really to improve and try to optimize rehabilitation interventions for cerebellar ataxia. In my laboratory, we use a combination of computational and behavioral methods. Computational modeling is very useful because it allows us to generate hypotheses of mechanisms of sensory motor control, which we can use to simulate or predict behavioral outcomes of those mechanisms and what it would look like if those mechanisms are disrupted. We can then use behavioral methods to compare the behavior of individuals with neurological damage to see how it compares to our model simulations. For behavioral analysis, we have two main pieces of equipment in the lab. The first is a three-dimensional motion capture system. You might be familiar with these if you've ever seen a documentary about how they make certain video games or action movies. These systems have markers that are placed on anatomical locations on the body and cameras that strobe infrared light. That infrared light is reflected off those markers, and the cameras allow us to really precisely record movement. We can integrate motion capture with virtual reality. This allows us to manipulate what people see about their movements. The other piece of equipment we have in the lab is an exoskeleton robotic device for the arm. Robotic devices are also helpful in manipulating visual feedback about movement. But the advantage of robotic devices is that they also allow us to manipulate something called haptics. Haptics describe how a movement feels. By manipulating visual feedback and haptic feedback, we can create virtual environments and see how patients with neurological damage learn to incorporate those new environments. I think that the main impact of my work for clinicians is that it shows how an in-depth understanding of the mechanisms that underlie movement disorders can be really helpful in designing therapeutic interventions. Mechanism is often considered important for understanding impairment. And while it can be very difficult to learn or train someone away from an impairment, it is possible to learn or train someone toward a functional compensation. Understanding the mechanism underlying an impairment can be really helpful to tailor and optimize the intervention to navigate that pathway towards functional compensation.